Okay, great. So the topic of my presentation is devoted to bio-inspired random networks, and it was done in collaboration with my colleagues from my department, Dr. Ayelet Lessman, who works in mechanobiology, and her lab engineer, Dr. Oren Checheyan. And the first part of this work was done by my PhD student Daria Arlova and Dr. Artem Panchenko, who was a postdoctoral student of mine, and now he works in Germany in the University of Erlangen-Nuremberg. The work was motivated by the work of mechanobiologists. They are interested in study of interaction between the biological cells, and we all know that, maybe not all of, but biologists know that biological cells are embedded into the special matrix called extracellular matrix, which is basically a network constructed from the proteins, and its properties depend on the part of the body where these cells are. For example, the cells in the brain are very different from the cells of the bone, and that's why the properties of the extracellular matrix is also different. So if biologists want to mimic this surrounding the media to study the interaction between the cells, they need to develop some, to make some media which is similar, which has a similar properties as the natural extracellular matrix. And usually they use the biological gels such as hydrogels made of fibrin or protein. And this protein can be very different, as I said, and they need to know how much protein and what type of the protein to put into the mixture to have some special properties, elastic properties as well, for the gel. So we were interested in how the microstructure of the network is connected with the effective elastic properties of this network. And another question that we were interested in is how to simulate the discrete model of the network by the continuum model, such that to consider the interaction between a lot of cells, because if you will try to use the one-to-one discrete model, it will take a lot of computational time. And usually biologists, biomechanists can consider only interaction between two cells, not more, because it's hard to simulate large systems. But if we can make a transition from the real extracellular matrix or hydrogel to some discrete computational model, and then from this discrete computational model to the continuous one, we could have a large computational effort and simulate large systems. So our idea was to consider some area of interest and develop a procedure to describe its effective properties. In order to do this, we used the Easy PBC plugin with Abacus. This plugin was developed by Sadi Komari, who invented it actually for the composites that have several phases. And we applied it to the networks, random networks. The only problem is that in order to use this, you need to have the points, the same points on left and right boundary and on bottom and upper boundary. So we managed to construct the network such that to fulfill this condition. And also the problem was that his plugin worked only for initial initial configuration of the material, and we considered the process of elongation of the material and how the properties are changed in quasi-dynamics, I would say. So we developed the plugin to work continuously during the stretching, the loading. The next question was how to choose the area of interest, how to choose the representative volume element. So we considered different sizes of the networks for the cubic elements, and in a three-dimensional case we obtained the following result. After 300 micrometers the Young modulus converges, so there is no sense to, it doesn't make sense to consider the larger volumes. And also you can see that starting from this point the computational time grows exponentially, so it takes a lot of computational resources to consider the larger cases, but we need to consider a lot of, we need to do a lot of calculations, because if we consider the random network, we need to take the statistics into account. So in all our experiments, we considered at least 10 different networks with the same parameters but different topology, and then we made the averaging. So how we constructed the network? As I said, we need the regular boundaries. So initially we constructed the cube that have a regular network, and then we added a random shift to the random displacements to the points that are inside of the cube, and then we provided the Delaunay triangulation to remodel or remesh all the network, and finally we obtained the random network with regular nodes on the boundaries. The next question was how to choose the proper connectivity, because we know that actually first, what is connectivity? Connectivity is the average number of the connections for each node of the network. And we know that in real hydrogels, the connectivity is not so big. It's about 3 to 5, something like that. So we had the connectivity of, well, much larger connectivity, about 8 as far as I remember. And here you can see that we tried to play with two different networks. One has highly connected points, C connectivity equal to 7, and then we deleted some elements randomly. We updated the algorithm such that to avoid the elements that are not connected to any other nodes, and eventually we selected the network with a connectivity equal to 4. So we consider two cases, high connectivity and a low connectivity. As a result, we have a realistic representation of extracellular matrix. It's a discrete finite element model and the continuum finite element model. And you can see that, for example, if we have a discrete model with discrete representation, we need to use almost 50,000 elements, and the corresponding continuum model has just less than 7,000 elements, and it can be obviously calculated much faster. But yes, of course, the elements are different, because in case of discrete network we use the beam elements, and here we use hexahedral elements, but still the calculations with continuum models are much faster. Then we need to select the model for the individual fiber, And we used the rabbit collagen patellar tendon fiber as a reference. And then we looked to the strain-stress curve and noticed that it is similar to the behavior of hyperelastic materials. So we used them to approximate the test data, Ogden material, Yo material, and Marlow. And we noticed that Ogden model with third order approximates the test data better, and we chose it for the further calculations. Then the idea of homogenization is the following: we stretch our material in one direction, and this allows us to obtain the elastic moduli C11, C21, and C31. Then, after we have the finite deformation in one direction, we apply the small deformations, small strains in two orthogonal directions and of the cube, and that allows us to obtain nine elastic constants, of which actually only 6 are independent. Why I think that only 6 are independent? Because initially our medium is isotropic, obviously, because it's random. But if we apply the stretching, it's like apply the induced anisotropy, and we still have 3 orthogonal planes of symmetry, which means that we actually have the orthotropic material, 3-dimensional. 3-dimensional orthotropic material has 9 independent elastic constants. We don't consider the constants C44, C55, and C66. We are interested just in this, and that's why we use them to obtain the moduli and the Poisson ratios. We decided to concentrate on these parameters, the engineering constants. Okay. Then we do the following thing. We consider stretching of the discrete network in one direction, and then we apply stress-strain curve. Then we use this test data, not for individual fiber, but for network already, to approximate it with the hyperelastic materials. And in this case, we see that most suitable model is a neo-Hookean model. Why neo-Hookean? Because we use all these materials with the continuum models that were chosen before. And we can see that whether the test data gives the following stress-strain curves, the others are laying a little bit higher, those that approximate this curve. But if we look to the elastic moduli in 3 directions, the closest one to the test data is the neo-Hookean model. So we choose this model as a best approximation for the discrete model in case of highly connected model, and these are these parameters. Then we decided to look to the loosely connected model homogenization, and in this case we have some distinguishes. First, you can see that stress-stretch curve is not monotonic. At some point, we see that it becomes unstable, and if we consider the real experiments with the collagen network, we can see that actually this is similar to them, but in case of real materials, this is a rupture. In our case, it is not rupture, but rather remodeling of the network. but still we decided to choose this 15% stretch for our experiments, and then we do the same thing that we have done before for the highly connected networks. We try to approximate it with continuum models, those that are stable at this regime. And in that case, the best approximation is given by the first order Ogden model with the following parameters. Here you can see comparison of these two models, the discrete one and of the continuum one, and you can see that indeed the field of displacements is very similar. So most probably this continuum model will work good for us if we want to describe the networks on a higher levels. But actually we didn't check this before. This is our future plan to check how indeed the continuum models works with to describe the interaction between the networks, between the cells, excuse me, and how the discrete model work with the cells and how similar they are one to each other. So the preliminary results are the following. The hyperelastic models can, no. How much time I have?
I think
okay, so the conclusions are that hyperelastic models can describe the discrete networks, and very accurately. The problem is that for different connectivities and for different collagen types, you need to choose the proper model, and that can be different models and different parameters. Okay, very briefly, the other work that is similar to this one, but a little bit different aspect. It was also motivated by the intercellular interaction, but consider dynamics. So we know that cells are interacting one to each other, but usually this interaction is considered as a static one. And our question is, what if the cells are interacting dynamically? Maybe they send the impulses, and this is much more effective than try to stretch all the network, all the surrounding around you. So we decided to build a particle model and simulate its behavior for different durations of the impulse which is sent from the active cell to the passive cell. This is a general idea. I will skip our experiments with the one fiber. This is just, I just want to show that we play with the following impulse, half sine impulse, and you can understand that this impulse can travel from the active cell to the passive cell, reflect and go back. So we are interested of how much signal we obtain on the reflected size. And obviously, we understand that if the impulse is short, that on the passive end, and the passive end, the impulse is doubled, and the maximum ratio between the force that we have on the passive end is twice higher than on the which was sent from the active end. And the shorter the impulse is, the higher is the ratio between the dynamic loading and the static loading. This is what we need to know about the impulses that are sent to the longitudinal chain, let's say. But what about the network? It turns out that in the network, there are some different laws. We have the active cell, we have the passive cell, we have a network, and the network consists of the fibers, and the fibers consist on mass on the springs. This is basically our model, and you can see that we considered a lot of particles and a lot of links between them. And it turns out that in case of the network, we also have some optima, but that is not like in the one-dimensional chain. In one-dimensional chain, if the impulse was short, then dynamic interaction was more preferable to the static one. In case of the network, the shorter is the impulse, the lower is the signal. So the network damps the signal and doesn't allow it to propagate. For the case of it just arrests the signal. And you can see that we are going almost to zero. And if the impulse is long, then we obtain almost one. So the same signal as it starts on the active end. In case of, if we consider the signal that we obtain on the passive end in comparison with that we have on the active cell, the rule is almost very similar. If the signal is short, then we don't notice the signal, and if it is long, then we have one-to-one correspondence, not two-to-one like we have before. How does it look in case of simulations? First, I would like to show the short signal. And you can see that it is arrested and doesn't propagate through the network. So we can see some perturbations, but the passive cell doesn't feel the signal. However, if we optimize the impulse, then the network allows this propagation, and we can see that obviously the second network feels the signal and does it very well. I also would like to mention that in the further in the previous experiments we considered the static positions for the left cell and for the right cell, so the boundaries were fixed. But if we let the active cell to move and the passive cell to move, then we also can see that still we have very similar behavior, and the passive cell, although it can rotate and that this decrease the signal a little bit, but still it feels the signal that was sent before. Here you can see some plots that show the signal from the active cell A and from the passive cell P, and you can see that for different durations the maxima are different. Maxima between the signals is different. Okay, I think I can stop on this to save the time. So, the last thing is dynamic. We see versus static propagation, and here we investigated how far the signal can propagate in the network in comparison with the static case, and we also obtain some maxima for the duration impulse. So the conclusions are that our simulation indicate that the cells can interact one to each other dynamically, and this works both for the fixed and free cells. We also introduced the limit until the impulse can be considered as a short one, and this is the following number, where tau in is the rate between the impulse duration and the time of signal propagation in the network. And also it's worth to mention that here I didn't show the experiments that include viscosity in the model, but we did such experiments, and we noticed that viscosity doesn't affect much on the signal propagation, and still we have the similar behavior. That's it. Thank you for your attention.